Click for IB Biology. We're looking at the structure of polypeptide chains. And now we're going to take a look at first at what are known as the tertiary structures. And then following that, we'll actually go on and take a final look at the quaternary structures as well. The PDF of this presentation is in the description. You can download that and annotate with your own notes. And there's also a quiz in there, of course, where you can check your understanding of your learning. The first thing to emphasize is that when we're discussing tertiary structures, we are dealing with a single polypeptide. This single polypeptide has, of course, folded up into perhaps an alpha helix or a beta pleated sheet, or indeed it is possible that the polypeptide chain is so long that sections of it are a helix and other sections are beta pleated sheets, and indeed that can repeat depending upon how long the polypeptide chain is. Remember, this is a single polypeptide chain. To illustrate this idea, I'm showing here on the left-hand side a single polypeptide chain that has an alpha helix region here, and then this links through a few amino acids to another alpha helix. This linking region is called an open loop, or sometimes is called a linker group. The second helix then gives way to a beta pleated sheet that seems to be attached back to the helix, and then at the other end another open loop or linker group attached back to the alpha helix. So the important thing to understand is that this is a single long polypeptide chain, a continuous chain of amino acids coiling into alpha helices, beta pleated sheets, short sections of open loop amino acids, and joins up into this new and more complex shape. Now, the reason why there are these twists and turns and folds to create this more complex shape is because of the interaction of the R groups sticking out of the chain, like so. Now, you'll recall from the variety of R groups that some of them are hydrophilic, or we could describe them as polar. They will form positive charges, and some of them will form negative charges. And it's possible that the different R groups, let's say that this one here is positive, might find itself attracted to this one over here, which is negative. And this causes a further twist and distortion in the chain of the polypeptide. Equally, there is an instance in which we have non-polar hydrophobic chains which are based on the idea of a covalent bond. Now, this is an unusual one in which let's say we have the polypeptide chains here. That could be an alpha helix or a beta plated sheet, doesn't really matter. And here we have an alpha helix and a beta plated sheet. But the amino acid sticking out, part of its R group is sulfur to hydrogen, and on this side hydrogen to sulfur. And what happens is the two hydrogens are removed and the sulfurs linked together. So if I just redraw that, and we have sulfur bonded to sulfur, and this is called the disulfide bond. Disulfide bridge is another name for it. Really worthwhile example learning. And this causes, of course, a very stable structure to emerge in the overall tertiary structure of this polypeptide. So the chain of amino acids is the primary structure. the alpha helixes and beta plated sheets, that's secondary structures largely due to hydrogen bonding. The tertiary structure is a consequence, yes, of hydrogen bonding, but more particularly of the interaction of R groups, of charged ones, and of the polar, non-polar ones with covalent bonds. And of course, this is just a reminder of the variety of R groups and the potential for the kind of bonding which might occur. Students are not required to know the R groups, and you are not required to know the details of the interactions in the folding of R groups. What you are supposed to know is an appreciation that this is causing the formation of a very particular shape. And with proteins, shape very much dictates function. Shape and function are related, and this will be the application that we make as we go on to consider the different proteins and their roles in biology. We're thinking about the protein shape telling us about the protein function, and that comes about because of primary folding into secondary, folding into tertiary shapes. But there is the possibility, quite commonly, that we have multiple polypeptides. So this would mean that we would have a tertiary structure coming up and combining with another tertiary structure, possibly 2, possibly 3, possibly more, often referred to as subunits, each one being a polypeptide chain, each one having a tertiary structure. And these groups are held together by ionic hydrogen bonds, non-covalent, or indeed once more by the covalent disulfide bridges. And this leads to, of course, an altogether higher level of protein shape and therefore function. In the image that you can see here, we have hemoglobin. Hemoglobin is known as a tetramer, which you will see when you study gas exchange. We'll consider the structure of that on the higher level course. And this is tetramer because it has 1, 2, 3, 4 subunits, each one a tertiary structure, and they're held together by various bonds in there to create the single quaternary structure of hemoglobin. Then just one final point to make about these proteins is that the quaternary proteins are often found in combination with non-metals, with metals, and in particular metals from the transition series of the periodic table. And those metals are ones that we often associate with catalytic activity. In the case of hemoglobin, we have the iron at the center of the heme group, but you could equally find other ones such as cobalt. The combination of a biological protein molecule with a metal is called conjugation. And this is particularly the case with enzyme molecules, which are often conjugated with transition metals. And that tells us something about their function as organic catalysts. But that will be covered in that section of the course. The links which are showing now are leading us on to looking at the idea of shape and function. There's a subscription button if you would like to subscribe to the site, and a link back to the playlist so that you can find the other protein videos.